Hello, good afternoon, and I'm quite happy that you can join us for this webinar on integrated photonics in health. Today we have a program led by TNO, one of the largest RTOs in the Netherlands, with a very large program in photonics and a very large program in health. So I look forward to interesting presentations. We cast the net a bit wider today because it's not only about the applications but also how you can make this actually scalable and at the right performance and quality. But you will hear that in a moment. I'm Karel de Vries. I'm from Photon Delta and I will lead you through our program. But before I do that, I would like to ask everybody in the call to mute their microphones if possible until the last moment, because we will record a meeting, we'll make the material available, but we also have questions and sessions at the end. And the question session at the end, Peter van Arkel, our CTO, will gather in all the questions and ask questions to the panel. So that please put your questions at the end or during the presentations in the chat. All right, having said that, welcome again. I will start with introducing Photon Delta and why integrated photonics is such a good idea for healthcare, what type of things we have, and why it's a good idea to be in this vicinity working with our ecosystem because of the strengths that it has. Then we have 4 pitches, and we'll introduce them in a moment, but first start with Photon Delta. Photon Delta is an ecosystem accelerator, a business accelerator. We work with a large ecosystem of integrated photonics company and R&D organizations, and we have an organization ourselves, and it is most well known for the fact that we have gotten a so-called growth fund grant from the government of 1.1 billion, which we use to develop technology and the ecosystem and business. But besides that, we also work on international EU relationships. We do a lot of market development and talent development. And last but not least, we have a startup support team and a seed investment organization with and an investment fund that we use to support very early phase companies. We have a number in the meantime. We would like to expand that number considerably still, so this is one way to get there. What we also want is not only to have companies that do something with photonics, we want to be able to have a complete supply chain. So from designing the photonic chip to the wafer production of it, to the packaging of it, and of course the integration into modules and final solutions, because that is what customers at the end use. We do that with the companies, but we also do that with a large number of research and development partners, and TNO is one of the largest of those. For those who are not yet that familiar with integrated photonics, but I'm more interested in what you can do in health, what is so great about integrated photonics? Integrated photonics essentially integrates optical functions that exist nowadays with using lenses and all kinds of large and mostly bulky optical components, but it put it in a chip. And that chip resembles what happens to the semiconductor industry. It is essentially also produced in the same way as semiconductors are made, but instead of electrons, we're using photons. And it has a number of benefits. In datacom, it allows all the hyperscalers. In healthcare, it allows totally new sensing solutions. But the other thing is, and that's very similar to what happened to the semiconductor industry, once you put it in a chip, it becomes small, it reduces the amount of power that you need, and you can make it at scale, and that means that you can make a lot of them for a fairly low price. That's the area that we are developing. Now, in healthcare, you see a broad range of sensing solutions where you can use it for, including volume applications like wearables and point-of-care solutions. When I started in integrated photonics in 2019, there was actually one company already active in the field making fiber break grading sensors, and the rest was still working on ideas. In the meantime, I can say we have a fairly complete set of photonic technologies, and some of which people said could never be integrated in a chip, that are in the meantime being integrated in a chip, already integrated in a startup company. So In the meantime, we get to the point you say, if it's worthwhile business-wise, you can put it in a chip. Now, different healthcare solutions require different type of chips and solutions. So you have point of care that have got typically higher volume applications, but a general practitioner wants something real-time, fast, and easy to use. On the other hand, we have analytics and bioreactors and stuff like that. That's more industrial in that sense. They like the higher productivity and the lower cost. Laboratories and hospitals have various things, faster, better testing with biochips, for instance, specialized testing or monitoring, which is enabled by the fact that things are small and low power, and in that sense wireless. Then we have the personal stuff. You already know a number of functions that are optically enabled in your smartwatch, but this is only the beginning. There is a lot more to come, and there, of course, it has to be light, easy to use, and also very affordable. And home tracking is something in the middle, and there you have the solution. It has to be reliable, has to be able to interface with your doctor, but it has to be very easy to use. So, we enable a lot of possibilities. Why should you come this direction? We are, within the Photon Delta ecosystem, a global hotspot for integrated photonics. There's a lot of knowledge in this region about both photonics and healthcare and semiconductors and AI, because there's all of that is needed to make solutions. There's a complete supply chain, so we can not only think about it, but also make it. There's extensive support available in the Brainport region, many institutes, our ecosystem, the access to startup support, our fund, but also we link with many VCs that can enable new solutions. And all in all, we want to speed up the process from technology to market-driven solutions. So we sincerely hope that after this session you are inspired to connect to us and take the next step into new great solutions. Thank you very much. I'll move on now to introducing the next speaker. And you might know Natalia because she has been moderator in previous webinars before, but besides that she is also a great photonics and healthcare researcher herself, and has a long history in Philips, ASML, and also in TNO in doing both. Natalia, take it away. Show us what is possible.
Good afternoon, and welcome everybody to the wonderful area of biosensing, where technology is actually delivering impact. The speakers of today we represent, Dutch organization for applied research with over 90 years of experience working with government, with industry, and for industry, delivering societal impact. We as TNO, we are over 4,000 experts, and TNO as organization is structured in 6 units. The speakers today, we represent the unit high-tech industry, working for the high-tech applications, in close collaboration with the TNO unit Health and Work and TNO unit Defense, Safety and Security. And my talk will touch our collaboration in depth. Now, some of us represent, Whole Center in Eindhoven, and we are hosting this event today. TNO Whole Center is a center for open innovation and with really expertise covering different type of integration, from hybrid and printed electronics to chip integration, thin film technologies, as well as photonic integration circuitry, integration of photonics, and most importantly, integration into devices, medical devices, wearables, and sensors. When it comes to medical devices, pushing innovation and bringing it really to the market for medtech or diagnostics, it has been and it is a systemic challenge. And what industry are not really saying loud, but what I can say as an industry expert is that brilliant sensor technology by itself is not enough. So there are many barriers that really prohibit the innovation coming from the lab to the field, to the market. And these barriers, in particular in biosensors, include very scarce and different type of expertise, sensors, technology, biofunctionalization, fluidics, electronics, integration that is needed. In any case, it's very high integration risk assembling something from the lab components and moving to the high TRL level, eventually to the product. And also one of the hurdles is lack of the access to pilot lines and to production and supply chain. Now, to overcome those hurdles, we at the Whole Center, we have been working with industry and for industry over more than a decade, and helping our industrial partners to accelerate the innovation and bring it to the market. And a few examples that I can mention is vital signs wearables in close collaboration with Philips and companies like Shinetsu, DuPont, Henkel, Lohmann. Currently, we are pushing the limits of wearable ultrasound in collaboration with Philips and Cyber. Smart wound carrier is again the new area where we are working with industry. And we are actually building upon this foundation and success. And nowadays we are pushing the boundaries of biosensors where integrated photonics really can play a very strong and important role. Now, sensing is becoming to be really a strategic layer, not only for healthcare, but also for defense, safety, security, and space exploration. So think about diagnostics at GP office, at home, point of care. Think about resilience and combat readiness of the military NATO forces. And think about space exploration missions, discovering origin of life, or also monitoring health and wellness of astronauts. Now, the markets in health and well-being, CBRN, chemical, biological defense, as well as space missions, these markets are not only large, they also represent really strategic areas for Europe, for European health resilience, for European defense and security, as well as overall technological stability and superiority in Europe. So how we are addressing this, and I'll give a few examples today in this talk. One is, together with TNO CBRN, Defense Safety Security for Chemical and Biological Threats, we are working on so-called dual-use biosensing. So the targets are known. These are high-priority pathogens and toxins. And what is really needed here is a fieldable detector. So the fieldable detector that can be taken in the field, it's rugged, yet it should provide sensitivity better than current assays, real-time data, multiplexing, and the cost should be also accessible. And the surveillance of such a biosensor will be done then in different matrices, different media, in soil, in water, as well as in biological media such as blood. And again, I emphasize this is so-called dual-use biosensor, which is addressing the needs of defense as well as of health. And that is important to drive the volumes in solutions offered by integrated photonics. Now, to address this need, TNO, together with its industrial partner, Microneat, we developed innovative platform which combines CRISPR-Cas lump amplification, and it is deployed on a scalable platform of molecular diagnostics offered by Microneat. CRISPR, it offers molecular specificity, and we combine it with a lump amplification in one reaction, so they deliver specificity and sensitivity, multiplexing, and we deploy that on a platform of Microneat, which is, in a nutshell, is a disposable cartridge where analyte and reagents come, and a reusable diagnostic device where the thermal cycle is happening to enable lump reaction, and where fluorescence detection is happening to detect the fluorescence label. So we, TNO and Microneat, we developed this technology. We tested it at biological safety labs class 3 at TNO CBRN facilities. And the data show consistent performance of Microneat platform. The data show very high specificity of this test, and we are very sensitive. We can detect pathogens, in this particular case, Bacillus anthracis, at the level of 4.5 DNA copies in a sample. If you would like to follow up on this, contact the business developer, Freek. Now, photonics And integrated photonics actually offers a new chapter in the biosensing, because photonics offers a compact device, rugged device, where you can combine multiple functions together. And in particular, we'll speak today about evanescent field for biosensing, and Peter will take this technology further later today. Evanescent field peak-based biosensing is really building upon over 20 years of expertise. Here in the Netherlands, it started in Messeplaats in Twente. And finally, we are reaching the opportunity, the time where this technology is really hitting the market. And let's not forget, this is a unique opportunity to build the ecosystem and explore this technology here in Europe. Now, why I'm saying that? Because in the US, actually, the company Genolite already commercialized evanescent field biosensor based on PEAK, and they deployed in hospital for hospital applications, but that's US. And as I already mentioned from the beginning, the area of healthcare and diagnostics, the area of sensing for defense, safety, security, that is really of strategic importance for Europe. Europe, and in particular here in the Netherlands, we have the company Surfix Diagnostics, who built upon years of research and collaboration in their own labs, but also with Lyonics, integrated photonics foundry here in Twente. And Surfix is bringing to us ultra-sensitive molecular diagnostics platform based on PEAKs with their proprietary nanocoating technology with integrated microfluidics cartridge. Importantly, that Surfix tested their own technology versus a gold standard, and they show performance which is actually exceeding the gold standard diagnostic platform. And that offers a great perspective to nurture this technology and deploy to the market here in Europe. And TNO, Logan will speak about it later, TNO is collaborating with Surfix on optical coupling to enable this technology going to the market. Now, we as TNO, we have our own track record in integrated photonics evanescent field biosensors. For over 15 years at TNO, Peter Harmsma, who is a speaker today, he has been working on this technology, and the success is proven. TNO created a spin-off, Delta Life Science. It's a company that already brought to the market CE-marked evanescent field biosensor addressing life science applications. And that's really the proven success. Now, we have been using our TNO technology, evanescent field sensing, to address applications for defense. In particular, here I show an example of detection of a nerve agent, exposure of people to Novichok's glass components is lethal, and detection of whether the person was exposed to that can be done based on specific biomarker. Evanescent field sensing shows very high sensitivity. We got the results within the minutes, the concentrations which are clinically relevant. And as we speak, we are extending application of evanescent field technology to another use case. It's detection of high priority pathogens. Again, here we are addressing two domains. One is CBRN safety security, and another one pathogen detection for so-called zoonotic diseases that spread from animals to humans. And I show here just one example of binding kinetics of membrane protein is Bacillus anthracis and antibody, the data we recorded on Delta Life Science platform. Now, the main question is actually not whether evanescent field can be useful for biosensing, but how to deploy that in a fieldable format to address portability, rigidity of this technology. And there are two routes. One is a pluggable cartridge and the reader. And this technology is developed by Surfix, and Logan, the next speaker, will speak about it in more details. And another route is actually a fully integrated system with a laser detector and waveguide, everything integrated in one unit, and earlier Fix showed this solution. So the speakers of today will address both routes with the technology, and to summarize or to sketch a perspective, diagnostics, detection, surveillance. These are really strategic domains for healthcare, for defense, for space missions. Photonic-based biosensing can bring this technology to the next level. The key challenge is how to scale from lab to the field or from lab to industrial manufacturing. A few more challenges are there: biofunctionalization and sample preparation. We at the Netherlands, we have a very good position to build upon this expertise. We have Photon Delta, we have next-gen high-tech lab-on-a-chip ecosystems, and we, TNO colleagues, we are working with industry and for industry to bring photonics further. The next speaker will be Logan Dilly, and he will continue upon this topic. And if you're interested, please contact us, I hope you are, and reach out to our business developers. Thank you for being there.
Thank you very much, Natalia. I think I know the fact that you could have gone on with at least another hour of more examples, because it is in a fantastic field. But let me introduce Logan Dilly. Logan works in TNO at the northern part of the Netherlands in PATC in Enschede. He has factually a background in robotics, but in the meantime has switched to the biosensing applications, and as Natalia already indicated, making these devices in high volume, high quality, at affordable cost requires special packaging technologies, and he is going to tell us more about that. Take it away.
Thank you for the nice introduction, Carol. My topic today is scalable PIC packaging through element-tolerant coupling. In the slides, I will present our brand PITC, followed by the main topic at hand, optical coupling, and followed by the different applications and how each application needs its own optical coupling strategy. At the end, I will talk about the approach we took with PIC biosensing and how we are enabling it for high volume applications. PITC is a joint innovation center organized by Photon Delta, TNO, TU Eindhoven, and University of Twente. We work on 4 different technology programs, metrology being done at Eindhoven, silicon nitride technology based in Enschede, Twente, indium phosphide technology based in Eindhoven, and heterogeneous integration based in Nijmegen and Holt Center. If you look at the high-level process flow of photonics, you have the front-end process and the back-end process. In the front-end process, you have the wafer manufacturing, you have the chips that have the required functionality, and you do the wafer-level testing, and then it is transferred to the back-end process with the known good dies. At the back-end process, you do the electrical packaging first, have the PICs die bonded to the back substrate and wire bonded to the required tracks. Then you move it onto the final packaging, which is optical interconnect. This is where the PIC gets a chance to communicate with the external world. My topic today will be surrounding about the optical packaging and optical interconnection, especially looking at the fiber-to-chip coupling. If we look at the fiber-to-chip coupling, there is 2 different ways to couple the light to the chip, either through edge coupling or through surface coupling. Edge coupling is when you couple the light to the chip through the facet, and in the surface coupling, you couple the light on the top surface of your chip. Edge coupling has higher coupling efficiency and broader bandwidth compared to surface coupling, and also has a lower polarization-dependent losses compared to surface couplers. However, at the back end, it becomes a bit complicated with edge coupling, especially if you need polishing and also if the Moore-field diameter is too small. But it's not the case with the surface couplers, because the grating couplers are much more alignment tolerant, and also there is no need of a polishing step. In addition to that, surface couplers enables wafer level testing, so you can already test the wafers on wafers and find the known good dies before you actually package them. It's not possible. It's also now possible with edge coupling, now with an additional step of having a trench. We benchmarked various fiber-to-chip coupling techniques, and we see still the current state of the art for most prototyping is connecting it with the fiber. And we'll see the losses of about 1 dB with a 0.5 micrometer alignment tolerance for an edge coupling, and 3 to 6 dB with an alignment tolerance of plus or minus 1 micrometer for grating coupler. And some of the companies that provide these services are Fix, Meizu, and SQS. And there are also spot size converters from Lyonics, Ligentech, and WAPs that are provided by Team Photonics. And there is also some research done by Intel and Ixscale on bringing glass optical bridges for scalable solutions. There is also very high alignment tolerant photonic wire bonds that's been introduced by Vanguard and C2MI, and adiabatic couplers, which are providing lower coupling losses with a moderate alignment tolerance of about 2 micrometer. There are a couple of companies that are already worked and prototyped these techniques with mainly MIT and IBM. And there's also solutions in regards to micro-optics from Focuslight, Femtoprint, Senko, and Terramount, looking into CPO packages. And they can provide an alignment tolerance of about 30 micrometer with an optical losses of about 3 dB. And there's, at the end, we have also freeform couplers where there's a 2-photon polymerization technique introduced by Nanoscribe and Vanguard, where you can easily print your lenses on top of the PIC or a fiber array, and have the chance to play with the coupling loss and alignment tolerance. The best alignment tolerances so far it's proven with this technique is about 14 micrometer with 0.76 dB losses, and the best coupling loss and alignment tolerance that's been proven with it is about 0.5 dB and plus or minus 2 micrometers. If you look at silicon photonics, there are multiple applications at the moment. You have 5G future networks, data center, quantum, lab on chip, neural network, and quantum key distribution. Each of these applications require its own coupling strategy. Depends upon multiple parameters such as coupling losses, polarization dependent losses, packaging cost, expected production volumes, and alignment tolerance and ease of use. For the sake of simplicity, I just took 3 main parameters, which is coupling efficiency, cost, and alignment tolerance. Let's look at a couple of use cases where which one takes priority, and based on that, how the people solve the problems. In the case of SNSPD for a quantum processor, it is very vital that you capture every single photon, which means you needed a higher coupling efficiency, and they don't care about cost or alignment tolerance. So there is a solution that's been developed is from Munster University, where they used adiabatic coupler with freeform coupling, and they can demonstrate up to 0.4 dB of loss with plus or minus 2 micrometer alignment tolerance. If you look at the CPUs for the next generation data centers, there are people looking at coupling, a balance between coupling efficiency and cost, but they are okay with having a moderate alignment tolerance. So the best solution for them, best coupling strategy for them is adiabatic coupling. In that case, people are working at the moment on glass interposers and trying to have their photonic chips bonded to the glass interposer and guide the light through the polymer waveguides. Now, coming back to the biosensing approach, if you look at the current market with biosensors, there is always some kind of active alignment between the PIC in the cartridge and the optical unit in the readout instrument. Our approach is to completely eliminate this active alignment, and we believe that to truly scale and adopt biosensing in POC testing, we need a low-cost and alignment-tolerant passive solution. That's where we look at, and we looked at the benchmark, and we see that to have a low-cost and alignment-tolerant solution with a nominal coupling efficiency, micro-optics is the best solution. Micro-optics enables us to expand the beam on PIC side and on the fiber array side. By expanding the beam, it also gives an additional advantage of having the cartridge being insensitive to dust particles. We have started our work last year based on the paper from Peter O'Brien's group, We did multiple optical simulations, playing with coupling efficiency versus alignment tolerance, identifying different lens materials and lens attached materials, and we successfully started producing lenses in the lab scale through lithography and imprinting. And we are also doing extensive metrology to understand the process and to have a repeatable process for the future high volume productions. And we are currently in the step of characterizing these lenses, and we can talk more about that the next webinar. So to scale biosensing, we believe that we need to attain low-cost optical coupling. We believe that we can do it with imprinting process and lithography process. And to achieve the high alignment tolerant fiber-to-chip coupling, we can use the micro-optics for it. And to attain a pluggable technology with repeatable coupling loss and ease of use, we are working on multiple compliant mechanisms between the cartridge and the readout instrument. So with these three criteria being attained, I think we will be able to scale big biosensing. And in the meantime, I'll also thank our industry partners, Biolume and Surfix. Thank you for listening.
Thank you. You can see what it takes to really bring this to life. We'll continue with that because high volumes require more technologies in order to do that. So Ricardo will now continue with, using photonics to create other photonics, laser-induced forward transfer for PICs.
Yes.
Go ahead.
All right. Yes, thanks for the introduction. This is going to be the only talk probably that is not going to go into the detail of biosensing, but it's about how to create a platform on which you can host the biosensing functionalities, specifically using LIFT, laser-induced forward transfer, for photonics, and how this can solve some of the challenges in the heterogeneous integration of those components. As it was presented before, the photonic integrated circuit is a miniaturized integrated photonic system that has the advantage of have like an increased performance, better stability and reliability, and it's meant to be cost effective. However, when you think about how to create this photonic platform, if you look at the foundries and the market and the materials, there is a different variety there that can be used for that, ranging from 3.5s to silicon to new emerging thin film lithium niobate. So the question is like which one to choose for making this chip. The answer that we give is to do heterogeneous integration. So the idea of like combining the similar chips from different foundry, different materials into all one, because all of them comes with a lot of specific functionalities and they excel in specific domain, but none of them can cover all the full requirement that the system needs, which means being able to generate light, transport light, modulate light, as well as detect light. Indium phosphide or 3.5s in general are extremely good in generating the light and modulating. They are still quite expensive and not that well compatible with the CMOS technology, so looking at the semicon domain. On the other hand, silicon photonics and silicon nitride are way more comparable, so from the industrial perspective, they make more sense as a base platform. And they're also ideal for low propagation loss for the transport of light, but they are not that good for like generating the light or modulating it, or up to a certain extent with certain requirements. And then you have the more emerging material, class of material like thin film lithium niobate that are very good for modulation. And so combining all of them together in once is the direction to go, because you get the best of all the different worlds all in one. When you want to integrate, however, heterogeneously, there are a couple of challenges that comes and that needs to be tackled. All combined together also means that those different platforms and components, they need to be able to communicate between each other. So you need to have optical coupling. Light should pass from one component to the other, moving around and connect and whatnot. And for that one, you need a very specific, two requirements that are also like challenges at the same time. A very good alignment between the functional active part of this component with respect to the receiver or main platform, and also like the most pristine, clean coupling interface. You want to do the integration also at a very high integration speed to be economically reasonable as a technology. But also you need to be able to handle this new generation of very thin and fragile components called coupons, which comes in the very and most oddly shape, very thin, micron to submicron even, but very long millimeter and so forth. Looking at the spectrum of technologies available at different TRL on the market, you would ideally like to do monolithic integration, grow everything together. This works for some platform, but not for others. 3.5s on silicon still have too many defects in the growth. You can do wafer-to-wafer bonding. You grow them in two different platforms, and you just bond them together. But then you have to remove wafers, and the density of devices you can use, the usage of the wafer is also not optimized, so it will result in a very expensive epitaxial waste. You can do discrete die-to-wafer integration, so you fabricate the components, and one by one you take and you integrate. It's a bit of a slow process, it's iterative, and also like at the current status, the best pick-and-place machine cannot handle very thin components. The most promising technology now in the photonics domain is microtransfer printing, so you parallelize the integration using a dedicated stamp that picks up a certain number of components and prints all of them down on the other platform. The limitation of that, it is non-selective, and that it requires a little bit of, let's say, alignment between the pitch of the different elements that you have in the integration, the native, the receiver, and the stamp itself. Our approach to that is to use lift or laser die transfer. So essentially, a technology that uses like laser shot to integrate components and effectively pushing a component from one substrate to the other when they're put in a very close proximity between each other within a transfer gap. I'm running the animation, so it kind of like shows how the process works. Of course, it requires an alignment between the two platforms. It's a non-contact transfer technology. It's ideal if you have like topologies in there, and also for cleanness, and I will talk later why. The release of each component is very fast, few microseconds per die, so it can really lead to very high throughputs, and it's highly controlled. You don't damage the component when you transfer it. It is also extremely selective, so you can have the non-good die approach. You can transfer one component, not affect the neighboring one, and repitch densely populated components into like whatever other kind of design you want, and it's compatible with different substrate sizes. A key part of this technology is that it is a combination of a collective process and a selective process. So initially, our proprietary carrier picks up all the components at once that can be very densely populated and fabricated on the source wafer all together, and then from one single wafer source, you can then populate many different receiver wafers. So the scalability of this technology is extremely relevant there, and it can make like a very big economical impact when you look at components per hour or wafer per hour that you can populate. This is an example of our collective wafer scale pickup process of lasers. And I wanted just to highlight that our process, when it's semi-automated, it can typically pick up 90 to 95% of components. So you're not wasting component, or you're not leaving components behind, even when they're very densely packed, very millimeter-sized component with very tiny gap in between them. One of the very key advantage actually of our technology and the fact that you split the pickup with the selective release is that in the in-between, prior to release, you can do like a cleaning and activation step or whatever other processing to your components. This is very important, it's quite unique, and you can do it at the wafer level. So all the components can be treated, can be activated for specific type of bonding, for specific surface treatment that you want to have. And this is something that we notice in our case when we work, for instance, with coupons technology and tethers, that you can completely clean your surface, and that has a strong impact later on when you want to integrate them and keep the pristinity for the coupling. Our technology, it's also been like modified a little bit with the lasers and with our specific stack, as such that it can accommodate all the different kind of dimensions that those photonic components can come from, going from very tiny photodetectors to very big lasers. One of the key advantages is that it is not affecting neighboring die. So you can create your native source material wafer with many components, very densely populated, as you can see in the little sketch on the top right, and then selectively transfer only the green one, for instance, without affecting all the other and without having like very high energetic demand from the laser itself. We also showed beyond photonics that our technology can scale to even larger semiconductor chips, millimeter size, millimeter by millimeter, and we are looking also to push even beyond that. Of course, like accuracy is the other key challenge that you need to have there. We develop an acceptor technology that works in combination, and it's part of our process, that allows you to create an adhesive-less interface, meaning that when the component is integrated, it doesn't need an optical adhesive to be bonded to the receiver, and it's in effect connected via a direct bonding to the surface. And our acceptor technology is also like designed to work with most of the relevant photonics platform. As a matter of fact, we didn't find one that doesn't work yet. And it doesn't compromise any prefabricated photonic or electronic structure that you put on. So it's quite versatile. Here is a couple of examples of integrated photonic chips. I will not go through them, but you can see. Going from mechanical components to elongated one, but also to functional components like photo detectors on a functioning SOI peak for LiDAR and other applications that we explored. With this technology, in 2025, we proved some micron accuracy 1 sigma. And with the next generation that we are working on now, we already went to 500 nanometer 3 sigma values. And we have in our integration roadmap the ambition to go even further to 200 nanometer, and of course adapted more and more to any kind of component dimension that you want to have there, and in parallel scaling this from 2-inch to 4-inch and 6-inch source wafer, with the 6-inch potentially being the pilot line that TNO is opening here on the high-tech campus. One last slide on how to get to the 200 nanometer and beyond. Over the years, we work on different generation of tool that's been developed and improved, on which we validated the process, the throughput, the accuracy, and the scalability of the dimensions. And for this next step, we believe that combining synergetically the process development and the tool development is the key. So we are working on a new module, die-to-wafer module, R&D module for advanced packaging that will have a very advanced automation, mechatronics, and inline metrology in there, and also environmental control for the next generation of integration of chip. Yes, thank you. If you have any question, you can please just email me.
We'll do that in the chat. Thank you very much. Last speaker of today is Peter Harmsma. Peter himself is already quite well known in the photonics world, but he in fact started this by doing his promotion work at Ben Smith, which in himself is an icon in the integrated photonics world. He then proceeded to work in photonic companies like 3.5 and did biosensor research that led to the startup Delta, which we heard earlier in this talk. So I'm really looking forward to hear now about your next big thing in the use of integrated photonics for ultrasound transducers.
Thank you very much. So I will talk about integrated photonics for the detection of ultrasound, in short IPUTs, and the objective is to enhance the sensitivity and also the signal-to-noise ratio. We do this work at TNO, but we also, of course, do this work with our respected partners, especially those in the European project Mediput, where we work, amongst others, with Ligentech and VTT, and also from support from CITC. So medical ultrasound imaging, I guess it is widely known, very well known for pregnancy echoes, for example, but also in hospitals it's widely used for imaging cardiac diseases, liver diseases, but also sports injuries. So this is a very mature and widely used technology. However, what we would like to achieve still, or what the doctors would like, is even better images, because that makes your diagnosis more specific, more early and better, And ultimately, of course, the objective is to have better patient health and also lower recovery costs, healthcare costs, because the earlier the diagnosis is made, the faster the recovery. Now, the way ultrasound works, in a dispolite way of saying it, it's like, for photonics people, it's like an acoustic LiDAR. It's a bit disrespectful, but the way it works is essentially you have a transducer which emits a short sound pulse, typically in the megahertz range, into the human body. And this sound pulse reflects from organs, for example, like the heart. And then the reflection arrives back to the transducer, which does both emit and receive. And the key features here are that deep-lying features have only a weak acoustic reflection. So, of course, the deeper the organ, the weaker the reflection. So you would like to have a very low detection limit, so high signal-to-noise ratio. But there are also stronger reflections, so you also need a large dynamic range. Then, to have a high actual resolution, you need to have this pulse to be very short. And the short pulse means that you have a high bandwidth of your acoustic signal. And also for high lateral resolution, you like your acoustic wavelength to be small, meaning high frequency. So these are some key parameters, and they're also intertwined. Because if you go to high acoustic frequencies, that means you have stronger diffraction, and it means you have stronger attenuation by the human tissue. And that immediately feeds back to high signal-to-noise ratio and large dynamic range. So it's all connected. Now, traditional transducers are based on piezo materials. Piezo materials, they expand and contract depending on the voltage that you apply, or the other way around. And a key feature here is the noise equivalent pressure, so the smallest pressure signal that you can detect, which is ruled by, of course, the noise on the one hand, and the other hand, the sensitivity of your device. Now, when it comes to sensitivity, this is mainly determined by the piezo material properties, in particular those three that I've listed over there. When it comes to the noise, that is mainly determined by the electronic system, where especially the electric impedance of the piezo is of relevance. And these things are not independent. So when you try to boost your sensitivity and tweak around with your piezo material, immediately you start to see compromises in terms of noise. And this is the reason why piezo transducers they're very mature, but also the performance seems to cap at a certain level beyond which it is very difficult to enhance the performance even further, which is in the order of a half a Pascal at 1 megahertz. So how can we get the performance even better? So this is where integrated photonics comes in, at least in our vision. So what we do is we take in this case, a ring resonator, which sits on a buried oxide layer on a substrate, so a typical photonics platform, and we remove the substrate below the ring resonator, so that the ring resonator now sits on a membrane. And what happens is that the membrane deforms because of the acoustic signal, and as a result, the ring resonator waveguide will change in physical length, but also the waveguide cross-section and material effective index will change a bit. And as a result, there will be a change in the optical phase in this ring-shaped waveguide. And when you include this in a ring resonator or an interferometer, you can actually transfer that to an intensity which you can detect by means of a detector. Now, the key thing here is that the mechanical resonator, which is the membrane, and the optoelectronic readout, which is your system to read out the ring resonator, they are decoupled, unlike the way it is coupled in the traditional piezo material transducers. So we believe that this is the way how to boost the performance even further because of decoupling, sensitivity, and noise. Now, one thing you can do is you can do this with a ring resonator. So typically you can put a laser line at the edge of the resonance, and then when there's an acoustic signal, the resonance will shift. Whoops, a bit too fast. The resonance will shift, and you measure a change in transmission. You can also do this with an interferometer. We like interferometers with 3 outputs. I'll come to that in the next slide. And then, of course, upon a pressure signal, you can see the interferometer signal also shift. It's a little bit more difficult to detect. So what you can do is you can actually boost the performance by including more membranes in the same interferometer. So here I have 2, so I boost the sensitivity by 2, but you can also take 10 or 20. So this is where things become really interesting because you can enhance the sensitivity quite a bit. Now we like 3-port interferometers because the interferometer response actually has 3 unknowns, which are the amplitude, the phase, and the visibility. So the visibility is essentially, oops, things seems to go automatically. The visibility is essentially the modulation depth. And in order to know 3 parameters, you also need to measure 3 things. So you can solve for an equation with as many unknowns as many parameters. And what you can do is you can calculate the phase, which is the thing proportional to pressure, by the simple equation, provided that you have 3 outputs of your interferometer. And the key thing here is that now the phase is linear with pressure, and the dynamic range is quasi-infinite because you can just keep on counting those fringes. And linearity and dynamic range is something we really like. So when it comes to comparing ring resonators and MZIs, I have to go a bit quickly through this. When it comes to sensitivity, with ring resonators, sensitivity is in the same order of magnitude as what you have in typical piezo transducers. However, they use only a very small area. And the key thing is that you have to make effectively use of all this area within an acoustic wavelength. And this can only be done in a realistic system by means of these Mach-Zener interferometers with cascaded membranes. And when it comes to dynamic range, ring resonators are very limited here, whereas in MZIs, the dynamic range is virtually infinite. And when it comes to readout, with ring resonators, you need a pretty expensive narrow line width laser to account for phase noise issues, whereas MZIs are way more tolerant than that. What you also have to account for is multi-physics. So there's photonics involved, but there's also acoustics, mechanics, electronics, data processing. And just to give you a little flavor, here you see a list of 6 publications that we have published in literature, just about the modeling of these kind of membranes with waveguides. Because you really have to be able to predict what the performance of your device will be in order to achieve something that will work very well. Now, devices that we have made, we have not made them ourselves, so we work with our respected partners VTT, for example. So here you see these spiral-shaped MZI devices with membranes below. And as you can see in the top right picture is really a sea of devices, of which some are connected and some are not. Some dummies are included with the same type of spiral to make sure they have the same acoustic performance. And we also work with Ligentech, who also make interferometers, but also focus on ring resonators, because that's where they are very strong at. This was all made in the context of the Mediput program. Then the packaging and testing is done with the support of CITC. It's done partly at CITC and partly at TNO. You can see in the left picture the chip on the lower left with 2 fiber arrays, and it's all mounted on a black material, which is an acoustic backing to avoid acoustic reflections. And then we measure this at the water tank in The Hague because, well, to have a representative measurement, the human body is like, well, a bag of water with some things in it, so you have to test this in water. Then some key measurement data. You can see here the time response and frequency response of an MZI-based output made by VTT and of a ring resonator made by Ligentech. And here you can also see some performance in terms of noise equivalent pressure. Now I have to be a bit careful because noise equivalent pressure is comparing to other devices is often a apples and pears comparison. So I like to be a bit conservative here. Where we are now is roughly 1 order of magnitude improvement, whereas we see a good outlook for another order of magnitude, just, well, almost low-hanging fruit. So where we are going, we have the output building block, which is an MZI-based device. We can cascade many of those membranes into a single MZI. Every one of those will be an array, so that you have actually an array of receiver elements, and that will be all integrated in the probe head, which is something that a nurse can handle in the hospital. So in conclusion, the iPuts, integrated photonic ultrasound transducers, they promise to be a disruptive technology towards improved ultrasound imaging with demonstrated at least 1 order of magnitude, but the clear potential for 2 orders of magnitude improvement, where multiphysics modeling is really essential. And it's also essential to really know the application very well to make sure you design for the right parameters. And I have to be honest, the current TRL level that we are is around 3, maybe 4-ish. So it will take still quite a lot of time before this hits the market. Thank you very much.
Thanks, Peter. Please have a seat because we have now the questions that has been gathered by Peter during the presentations. And given the time, I guess, Peter, let's get straight into it. And
yeah, already in the
what
chat.
you hear.
Already in the chat, many thanks to all our speakers for the great presentations. And the first question we want to dive into is for Peter Harmsma. So you showed in the end of your slides that you're currently at TRL 3. You showed also a couple of new building blocks and directions where you want to go in the future. How do you see scaling in level of maturity towards the future?
Yeah, I think the key challenge is really in the fabrication. So when it comes to photonics, there are quite a number of mature foundries who can make your device according to your specification. Where the challenge is that if you look at the optimal configuration, we would like to step away a bit from the standard configuration, but move to non-standard stacks. But also the combination of MEMS and photonics is not so trivial. And the devices that we have made so far, they are made by removing the substrate below the membrane by doing a backside etch, which is a very, let's say, brute force approach. And actually you would like to go to a kind of a wafer bonding approach where we have only a very shallow cavity between below your membrane to make this more well defined. But also if you want to have many membranes very close together and you remove all that substrate stuff, there's almost nothing left. So it's not a very solid device anymore. So you have to go to this cavity-based approach, which is something that we are working on together with our partners. But that will be, in my opinion, a major step forward. Yeah.
All right, thank you very much for this clear answer. The next question for Ricardo. There were some questions whether you would be able to also transfer complex components and whether there are limitations to the complexity and also in terms of damaging them.
So we are already working on that. So today the presentation was focused on coupons itself, thin elongated components, but we have been also working and in not only in photonics domain, other domain in more complex system, different thicknesses or pre-integrated components also all together. To answer the part about damaging them, it depends on the type of damage, of course. Our proprietary carrier is designed as such that it essentially prevents that any of the energy that comes from the laser it gets to the device, so you don't damage the device. Now, if you want to be more specific, maybe like in biosensing, if you have like some biofunctionalization and whatnot, then maybe there you need to be a little bit more careful and you need to adapt a little bit your process in a way. But for standard components, for mechanical, thermal stability and let's say photonic electronic properties, typically they don't get damaged.
Okay, thank you very much.
Yeah, you're welcome.
So for Logan, the question on the coupling. So you have showed the assembly of different lenses. Can you tell a bit more about how you do this attachment and alignment?
Yeah, we have the alignment markers printed on our lenses. So it is also when you have the wafers ready, we would like to have some additional process on top of it. Having an alignment marker on the top of the wafer would give us an easy passive alignment between a MLA or a microlens assembly and a chip. That would it makes it much more easier just using just using your camera optics and then identifying these alignment markers and aligning your chip is very simple.
Okay, thank you very much. For Natalia, so in your slide you showed also a route towards mass producing evanescent field biosensing. And how do you see the critical bottlenecks towards scaling these applications?
Yeah, thank you very much for the question. Now, let me highlight that as I speak in my presentation, so biosensing for health and defense, that is becoming really a strategic area for Europe, for European health resilience, but also safety security. Photonics has a fantastic opportunity. And you, Photon Delta, you are doing great efforts in order to scale up, or in order to strengthen the ecosystem and enable this scaling up. A few other bottlenecks that are there is sample preparation, and therefore we are really eager to have the big biosensing companies on board working together with Photon Delta, because they have great experience in sample preparation. And another bottleneck that needs to be resolved, that is scalable biofunctionalization and biofunctionalization process that is actually compatible with integration of photonic components. And Logan has a long experience with that, working at FIX. So we need to tackle that together with the partners who know everything about biofunctionalization. So hence again, invitation to industries, industrial partners who are working on functionalizing surfaces together with photonic partners, how to make sure that biology and technology compatible, and that goes to market.
Thank you very much. My final question to Karel.
I wanted to close off.
Yes, I know, but I have one burning question for the last minute. So we heard all these great talks, and there is a lot of interest from the chat. And one of the questions there is, how can we collaborate, interact? Are there also opportunities to work together? And also, specifically the question regarding collaboration, for example, with academic partners in Europe.
Okay. You mean academic partners from Europe with our ecosystem? Okay. Yeah. That's a question where you can spend quite some time on, which I will not do. Photon Delta was essentially set up to basically enable more of these, not only in the Netherlands, but also within Europe. So I think that's, if there are people interested, please connect with us. It depends, the answer of what and how it can be done depends a bit on the exact cooperation. There are, of course, a multitude of European programs, and some of them we support, and some of them are already in the stage where they are being developed. If it is about using certain possibilities of facilities, then we can potentially enable that as well, depending a bit on what you want. And then, of course, if these things lead to startups, my personal hobby in that sense, we also have the possibility to enable setting up new companies. So, but it all starts with a conversation. I think that's the best answer you can give, and please do so.
Thank you.
And with that, I think we are at the end of our session. I would like to thank the audience for their attention, and the speakers for their interesting material, and I hope it has inspired you. So with that, have a good evening, and I hope to see you again.